Thank you very much, Alberto. Yeah, it's a pleasure to be here again. I think I've been presenting here at every single one of these for 19 years, is it now, or something like that. And it's always so rewarding and inspiring to see all of you here and to hopefully learn from us. And my job here is to present some of the basic research that's going on in Parkinson's from around the world. And I'll just give you a little snapshot of each. I'm not going to go into tremendous detail. I'm not going to use big, silly scientific terms if I can. And if I do, I'll try to explain. But what I want to tell you that in research, there's hope, right? But there's little hope without data. So you need the data. So if I can just go on here. Now, what I'm often asked is about stem cells. What's the latest in stem cells? Is that really going to be a therapy? And really, it's a kind of a cellular replacement therapy that Ben actually had a slide up earlier in his talk. And people are working around the world to optimize stem cell-based therapy for Parkinson's. And this particular group optimized patient-specific stem cells, right? So the idea is you're going to transplant human, something called induced pluripotent stem cells. Okay, a stem cell is a cell that can be induced to go into any kind of cell in the body. And that's what pluripotent means, multiple potent, it can go. But this is made from the patient's own skin cells, right? And it's promising as cell replacement therapy and more or less a personalized therapy because it's from your own skin cells. And the idea is that it replaces the dead, damaged cells that are deep down in the midbrain, the dopamine cells, with fresh, healthy cells, all right? And a key here is that in this case, since it's the patient's own skin cells, you don't need immunosuppressive drugs, right? So the immune system won't react against it like it would from stem cells from another patient or from another preparation. And how do they do this with these skin cells? They chemically turn them, apply chemicals in a dish and turn them into non-neuronal iPSCs into midbrain dopamine neurons, right? And then they take these cells. What this group did is they compare. They put these cells in culture, and you got to treat them. You let them mature in culture for different times. But you got to be careful. You got to take them at the right time. So they compared those taken at day 18 in culture from those taken at day 25 in culture. And they found that the timing, again, is critical for implantation into the brain to replace the dopamine cells. And they found improved motor function with these transplants, abundant cell survival. But the key that they found is that from those cells taken at day 18, process outgrowth from the cells that were grafted is a key factor in determining graft efficacy. And by efficacy, they mean functional improvement, okay? So if you look, I noticed that the arrow here doesn't work on the screen. So if you look on the screen, if you can see it on the bottom, there are two examples on the left and two examples on the right. So the 410 day 18 and 411 day 18 are taken from two different patients. And the brown staining that you see on the left of each panel is the dopamine cells that have been implanted into the area of the brain that's devoid of dopamine. And what you can see, you see those little stick-like brown things sticking out from them, kind of going off to the right in both the first two panels? Those are processes. Those are axons. Those are processes from the neurons. And what they're doing is they're going out and they're making contact with the cells in the striatum that is devoid of dopamine, okay? But the ones on the right, taken at day 25 in culture, you can see that while there's robust cell survival, right, there's a lot of brown staining, the processes don't go out, right? So what they found here is that also they did a gene expression analysis that revealed the certain stage-associated signatures of these developing cells, as they call them. So what they found is that at day 18, the genes that are in these cells that they're going to implant are primed, right? They're poised. They're ready to go out and differentiate and extend their processes and connect with the host brain and do good things and release dopamine. However, if you wait a week later, day 25, that stage is past. The genes then have stopped in differentiation, while the cells still survive very well, right? They don't extend processes anymore. They're like done. We don't want to stick out our arms anymore. So that's why this was a crucial step forward for potential clinical trials for human patients using their own skin cell-derived induced pluripotent stem cells. So another example of a potential molecularly based study is that there's a protein called NUR1. And again, scientists give silly names sometimes to chemicals and proteins, and it refers to nuclear receptor 1. It's a receptor in the brain, and it's in dopamine cells. And it's a master regulator of dopamine neurons, right? It's critical for development, maintenance, and overall health of dopamine cells. And in fact, in some Parkinson patients, levels of NUR1 are reduced, right? So now what this group was kind of a combination of a group from, I think it was from Harvard, and I forget where else, but from hundreds of candidate molecules, right? Researchers developed a new drug, again, 4A7C-301, they could have come up with a better name, probably, but they found that it had the strongest ability to activate this NUR1 and to reach brain tissue. So it was kind of two things that the key things here. So they found that this new activator of NUR1 protected midbrain dopamine cells in 2 animal models of Parkinson's, both a genetic and an environmental factor one, that it improved both motor symptoms, movement, gait, running, and also a non-motor symptom, that of smell deficiency in both of the animal models. Also, importantly, it inhibited brain inflammation, right? So a lot of times when you put in drugs into the brain, or even in these animal models, you'll get inflammation into the brain, which is a bad thing. And this inhibited that, so that's great. Also, it did not induce unwanted, these abnormal involuntary movements, these dyskinesias. So, and the underlying, and on the right there, you can see some examples again, the control lesion and the drug. So a control brain, if you look, the brown staining on the top is in the striatum, the target of dopamine cells from the midbrain, and the bottom is cells in the midbrain. If you lesion them in a model, you can see how the brown staining goes away and is diminished down in the bottom panel. However, with treatment with the drug, you can see that the brown is really robust again, it's up there, as well as more cells are evident in the substantia nigra, which houses the dopamine cells. So now, one puzzling thing was, why was smell improved, right? So this is not the pathway for smell. However, some of you may know that in the olfactory bulb, right at the front of the brain, humans, is the extension of the brain, the olfactory bulb, it also contains dopamine cells, right? So that could be the clue there, because NUR1, again, is important for dopamine neurons. So just to sum up, NUR1 then, right, is a promising molecular target for disease-modifying treatments of Parkinson's. What about the environment, right? Because I always hear about, what is in the environment that increases the risk factor? Well, environmental toxins and Parkinson's. That's kind of been in the news lately, and especially with burn pits, right, in Camp Lejeune and in other areas with veterans. So pesticides have long been linked to PD, but pinpointing, right, exactly which ones raise the risk of Parkinson's has proven really difficult. So this study, which was a joint project, and mainly from data gathered in agricultural fields in California, a group of UCLA and in India, but they combined epidemiology with toxic screening, right, in dopamine neurons derived from patients' induced pluripotent stem cells. So I told you about these induced pluripotent stem cells. So they screened so it's not only like they decided, well, this is a risk factor because they checked this population here. Now they're taking it back to the lab. They're looking to see if these were toxic in these special dopamine cells. So, from almost 300 pesticides that they looked at, they identified basically the top 10 that were directly toxic to dopamine neurons, and it varied. 4 insecticides, 3 herbicides, 3 fungicides. Unfortunately, most are still in use today, right? Only 2 are in use in Europe. So we're behind Europe in that sense. The herbicide trifluralin, which I had never heard of before, was the most toxic, and it impaired function of dopamines. The molecular mechanism of that seemed to involve the cell's energy producer, the thing called mitochondria, the powerhouse of the cell. And that's strongly been implicated in Parkinson's disease, mitochondrial dysfunction. And the kind of weird thing was that they noted that nothing unifies these pesticides, right? They're structurally distinct. They weren't classified in the same class of toxins or anything like that. So that was kind of a mystery. But however, this provides new clues about environmental toxins' role in Parkinson's. The next step is they're going to perform mechanistic studies on which specific biological pathways, right, are disrupted among PD patients who experience these pesticide exposures. Okay, and then finally, artificial intelligence, right? Good or bad? Well, here's hopefully a good example of artificial intelligence. So this is a study that was conducted, I think, through MIT, University of Rochester, Mayo Clinic, right? So all around the country, and which looked at sleeping problems and Parkinson's, right? Sleep disorders are a huge issue, as you know, in Parkinson's. And they have been historically associated with Parkinson's, breathing problems also. So what these investigators did is they developed an artificial intelligence, or an AI model, that can detect Parkinson's from reading a person's sleeping, breathing patterns, right? So they have a device in a bedroom that emits a low-power radio signal. It's reflected off the person's body and then analyzed. So I want to show you here. I know it's a little hard to see, but if you look in the upper left there, you'll see 2 examples, and there are people laying in a bed. And the one in the pink, where it says wireless signal, if you can read that. Anyways, there's a device on the wall. It's almost the size of a Wi-Fi router, right, that collects this signal, right, and the inputs are, it's sent to the inputs, which are, it's exhaling and different breathing patterns, right, and then that signal is sent to an artificial intelligence-based model, a so-called neural network that through some various algorithms generates the output, which then you can determine on the lower right if a person has Parkinson's, can it detect Parkinson's or not, and the severity of Parkinson's, according to this Parkinson's disease rating scale. So, but one of the key advantages here is that this signal is fed to this AI-based network to assess Parkinson's passively, right? So you're passive. You're at home. You're in your bed. It's all passive. And what they did here is they analyzed data from over 7,000 individuals. Most of those were controls, by the way, and one of the largest sleep studies ever conducted on PD. And the key here is that they found that this AI model can detect Parkinson's with very robust reliability and estimate both the severity and the progression in accordance with the UDP-RS scale, right? So this is kind of a proof of principle study that demonstrates the feasibility of something that's objective, right? So it doesn't depend on a patient self-reporting or the clinician, which is more kind of, it's not subjective, it's semi-subjective sometimes. It's non-invasive, it's low cost, and it's at home, right? So the advantage here is that, and I've heard people say that sometimes it's a problem, you only come in once or twice a year and see your physician for 20 minutes to an hour. Well, this could be set up to be used almost continuously, right, from at home. And those who live out in far-reaching rural areas, have disability with movements, can't get to the clinic downtown or something like that. I think this could be a really, really good advantage in this case. And so basically it's a potential new digital biomarker for Parkinson's where you can also track progression of the symptoms. So just to summarize, I told you that there are now personalized patient-specific stem cells which are being optimized for treatment of Parkinson's. And I think it's a real possibility in the coming years that stem cells, especially these patient-derived stem cells, will be put to good use. NUR1, this protein, is a promising target for development of new therapeutics. There's this new screening approach that combines epidemiology with toxicity in a dish, identifies specific pesticides linked to PD, and could really inform agricultural use in the future. And then finally, we have the potential application of artificial intelligence for really non-invasive Parkinson's healthcare. So thank you.